the trial, where they want the trial to be, and whether or not this is the side effects that they can endure.
All right, thank you. That would be the end of the Q&A session. Thank you so much,
Thank
Lynn
you.
Health. Next, let's welcome the last team of the day, Cogentis.
Hi, I'm Kent Werner. I'm a neurologist and a neuroscientist at Johns Hopkins, and I'm also co-founder of Cogentis Therapeutics, a multidisciplinary team that is uniquely poised to develop a cure for Alzheimer's. One of the most painful parts of my job is diagnosing someone with Alzheimer's. It destroys the mind, devastates the family. It's the most expensive healthcare problem in our society. And what's very frustrating is there's no cure that I can offer. And this is a crisis, because look around this room. 1 in 3 of us, by the time we turn 85, will be diagnosed with Alzheimer's. How do you get from a normal brain to an Alzheimer's brain on the right? Well, I want to pull your attention to an enzyme called CDK5. It has plenty of functions in the normal brain, but in the Alzheimer's brain, it's 10 times more active than it should be, over-modifying, phosphorylating proteins like tau and amyloid, creating toxic end products and inflammation. It kills brain cells, and pharma saw that as a great opportunity. Every pharma in town had a CDK5 program, but how do they do it in pharma? They inhibit the enzyme, and they're very good at it. Unfortunately, that was very toxic. All programs were scrapped, but fortunately, the 2 scientists who discovered CDK5 brought us a new way that pharma has never considered. Dr. Pant at the NIH, using discoveries made by Dr. Tsai, who's here in the audience, developed CT-526 to not inhibit CDK5, rather it protects it by keeping away abnormal activators like P25, normalizing its function. There's no toxicity. Give the mouse 100 times the dose, and it's still smiling. Especially the Alzheimer's mice, because they're the gray boxes, okay? And if you give them only 3 days of this drug, you'll see reductions in both the tau and amyloid I was telling you about. Biochemistry's nice, but also their cognition's improved in the maze. The advanced ones aren't falling off the rods anymore. And so what we have in multiple models now is evidence for the milestones preclinically and animal safety all the way through to proof of concept, telling a drug developer, a scientist, a doctor that it's time to get this to patients. And that's where Cogentis comes in. We keep collaborations with academia as well as with pharma to determine what are those key, de-risking, efficient, go, no-go experiments that get things done. We're not going to drill on with grants and interesting science. That's Dr. Tsai's job. Instead, we have developed a series of experiments that are specific go, no-go experiments designed to have value inflection with each result, giving us momentum for the next raise and giving us the opportunity to exit. You can see the cost there within 5 years. And what kind of exit? The field is hot. Here in town, pharma has paid over $500 million in the last few years for small preclinical companies just like ours. And this is not an easy task, and so you're constantly overcoming obstacles. The first one was when we locked down the intellectual property with the NIH. Next, Alzheimer's trials are notoriously slow and costly. Instead, we are first targeting a cousin disease, a rapidly progressive dementia called frontotemporal dementia, FTD. It's an orphan with the FDA, which means we save millions in regulatory costs. We get through the trials faster. Also importantly, industry expertise came to us. When they learned what we were doing, we were lucky to have Yvonne Angel. She's a peptide chemist for Wyeth and Affymax, among others. And Novartis has given Sanjeev Dohan special permission to work with us. He's put more than 20 drugs through the FDA. And I'd be remiss not to mention Sean Evans, who's in the back. He has built many large partnerships with Johns Hopkins and industry in his day job. And together we were able to put together a team that has brought home wins with the Neuro Startup Challenge at NIH, Mass Challenge, we were named winners here in Boston. And that's allowed us to deepen our network to people who have been there and done that. Bruce Morimoto at the top of the screen. He developed his own peptide for CNS with Alon Pharmaceuticals. Superstar AJ Verma, we both came from the same lab at Hopkins. He developed aducanumab for Biogen, which is currently in phase 3. So with your help and theirs, we will change the experience of aging. Thank you. I'm ready for questions.
Thank you. Now we have 3 minutes for questions.
Thank you for that presentation. Definitely a worthwhile effort to take on. So you mentioned that you're targeting, as you brought up, that Alzheimer's is a very progressive, long time. In that sense, one of the challenges also there's no diagnosis for early detection of Alzheimer at the time of the dementia or MCI. So to that extent, how or what confident you are that this new candidate can actually be able to, at early stages of the Alzheimer, even if you have mild cognitive impairment, to stop or reverse the progression of the disease before it's too late?
Really good question, and we are all over that. So first of all, the interesting thing is these mice that we treated, they would have passed for severe Alzheimer's. They already had the gait deficits. They're already showing the problems in the maze. And there's some interesting talk that we could discuss about lifting the fog, so to speak, because it's not, all is not lost when you're at MCI. Second, we are partnering with multiple biomarker companies for earlier detection, including blood test detection. In fact, some of our colleagues recently published a 90% sensitivity from the blood. So by doing those biomarkers and developing our own IP biomarkers for our own target, we'll get early signs of target engagement so we can keep pharma's interest, although they continuously tell us what they're interested in seeing, which is a good thing. Keeps us honest. Yes?
Besides the leading candidate, what other pipelines do you have?
So we had previously licensed a small molecule. We decided against it in the long run. We are looking at a couple other candidate peptides that are similar ways to approach the biology, but as far as we know, no other company has even touched this biology. And I think it's partly because pharma is very social, and they had problems with CDK5 in the past. But they are now calling us and asking what we're up to. So I think it's going in the right direction.
If you're successful, this is an extraordinary achievement. You list what I think are relatively low numbers for getting to market. No therapeutic I've ever heard of has made it through with their first molecule.
Yeah.
How are you going to find the money to get all the way to market?
Great. So first of all, those are not numbers to market. They're numbers to phase 1A and phase 1B. And that's a crucial point because our market is not the patients. I care about the patients. Pharma will do a far better job. And so they are our market, and we're constantly discussing this with them. And the money will come from SBIRs initially for seed type of things. And to carry us through, there's Create Bio Grant at NIH. That's a $5 million drug development grant. And they've already called us and said, hey, here's how you write our grant. This is a great thing to do. And so that, Ignite grants, and venture companies, I am not trying to hold on to all this equity and this job. We want to move it. And so if it takes venture early, I'll do it.
We have time for one last question.
What is the deliver's platform for the peptides to keep it stable to deliver until the target tissue?
That's a great question. So the stability right now, its half-life in the blood is less than 30 minutes. Shockingly, its half-life in the target tissue is several days. In other words, it sequesters itself by its own administration. And so that's with its current form. But we do have some lead optimization plans to modify the peptide that will increase its half-life, make it a little bit cheaper to produce, make it more potent. But we don't see that as a problem currently with the data that we have, but we still want to improve it.
All right, that would be the end of our Q&A session. Thank you so much, Cogentis. Now that all the teams have finished presenting, we are collecting scoring sheets from all the judges, and our team will be working on calculating the final